Dear learners, so far we have studied probiotics and prebiotics. Now in this section we will study synbiotics and postbiotics. Now synbiotics. Synbiotics are combinations of probiotics and prebiotics designed to improve the survival of ingested microorganisms and their colonization of the intestinal tract. Development in microbial research has led to the formation of synbiotics, which is a fusion of probiotics and prebiotic products and helps in enhancing the survival and the implementation of live microbial dietary supplements in the gut. The synergistic benefits are more efficiently promoted when both the probiotic and prebiotic work together in the living system. There is mounting scientific evidence that the symbiotic relationship between prebiotics and probiotics contributes significantly to health. Commercial interest in functional foods containing symbiotics has consistently increased due to awareness of the benefits of gut health, disease prevention and therapy. Research in this area is currently focused on developing new health promoting foods as well as on selecting new cultures demonstrating an enhanced ability to colonize the human gut along with their ability to digest new forms of prebiotics. Conventional trials and investigations have shown that various beneficiary effects of probiotics, prebiotics and synbiotics are much more effective than their unitary use known till date. Some of the common symbiotic compositions currently under study are shown in this table. Like fructooligosaccharides, they need bifidobacteria, inulin needs lactobacillus acidophilus, isomaltose oligosaccharide needs bifidobacteria, lactulose needs lactobacillus bulgaricus, lactose sucrose needs Xymonas mobilis, xylo-oligosaccharides needs bifidobacterium, galacto-oligosaccharides needs bifidobacterium longum, fructooligosaccharides need bifidobacterium bifidum, arabinoxylene and ariboxylene oligosaccharides they need bifidobacterium species, and resistant starch they work better with bacteroids and Eubacterium rectal. Dear students, you will be learning probiotics and prebiotics as separate units, but an attempt has been made in this module to give you an insight of how these probiotics and prebiotics will work synergistically. Now we shall study the most recently included term along with probiotics, that is postbiotics. Recent data suggests that bacterial products in the absence of viable organisms may have similar effects on signaling pathways and barrier function. These bacterial products are broadly categorized as postbiotics and can be defined as non-viable bacterial products or metabolic byproducts from probiotic microorganisms that have biological activity in the host. General postbiotics include bacterial metabolic byproducts such as bacteriocins, organic acids, ethanol, diacetyl, acetaldehyde and hydrogen peroxide, but it is also found that certain heat killed probiotics can also retain important bacterial structures that may exert biological activity in the host. Research shows that these metabolic products have a broad inhibitory property toward pathogenic microbes and therefore can be used as an alternative to antibiotics. Postbiotics are non-toxic, non-pathogenic and resistant to hydrolysis by mammalian enzymes as these are non-viable bacterial products or metabolic byproducts from probiotics. In some instances, postbiotics can also enhance barrier function against species like Saccharomyces boulardii and improve angiogenesis in vitro and in vivo in epithelial cells by activation of A2B1 integrin collagen receptors. Similar properties have also been identified in several other probiotic species of Bifidobacterium breve, Bifidobacterium lactis, Bifidobacterium infantis, Bacteriocides fragilis, Lactobacillus and Escherichia coli. Let us conclude. In this section, we have studied synbiotics, wherein prebiotics and probiotics work together, and we have also studied probiotics. The human gastrointestinal tract is colonized by a complex ecosystem of microorganisms. Intestinal bacteria are not only commensal, but they also undergo a symbiotic co-evaluation along with their host. Beneficial intestinal bacteria have numerous and important functions. For example, they produce various nutrients for their host, prevent infections caused by intestinal pathogens, and modulate a normal immunological response. Therefore, modification of the intestinal microbiota in order to achieve, restore and maintain favorable balance in the ecosystem and the activity of microorganisms present in the gastrointestinal tract is necessary for the improved health condition of the host. The introduction of probiotics, prebiotics or synbiotics into human diet is favorable for the intestinal microbiota. They may be consumed in the form of raw vegetables and fruits, fermented pickles or dairy products. Using bacterial products or metabolites remain an exciting yet largely unexplored therapeutic option. Heat-killed probiotics may also function in the broad sense as postbiotics. Postbiotics aim to mimic the beneficial therapeutic effects of probiotics while avoiding the risk of administering live microorganisms. Thank you.